this so that because there was at least one person that asked like that he can't, I felt bad Rabbi Spivak's having a class tonight. But so this way I can send it. So just FYI, like it's being recorded, whatever. And if I don't send you the recording within a week, then email me, I'll send it to you. And if you get booted from Wi-Fi or whatever, like, just come back to click the same link and come back and that's what I keep looking back and forth to make sure there's people, like if there are people, I'm letting them in. That's the story. All right. So without further ado, again, thank you, Dr. Lewis Schrager, so much. What a mensch. Ready?
I'm ready.
All right.
Before you start, I just want to say a couple words of introduction myself. I want to thank you, Zalman, for sort of engaging around this idea. We were speaking actually about Yaakov's Upsheren, and the idea popped in my head based about 3 weeks ago. I do some work here in Washington with the Jewish theater here called Theater J, and the artistic director, a fellow by the name of Adam Immerwar, learned of my background in infectious diseases and epidemiology, and wanted to get some insights on COVID so that he could make some decisions about the winter spring season of the theater. I shared some of my perspectives with him, and then he suggested that on the next monthly call of the Washington Regional Theater Leadership, which involves about 60 of the theaters in this area, that I engage in a Q&A with that group. And I heard it was considered fairly useful. Shared that with Zalman, and then we decided to engage with your community as well in this way. And I do hope it will be useful. I do want to emphasize that I will sometimes be giving opinions, and that also this is a moving target, and we're going to be learning more about this virus every day. Every day I learn more, every day that we all do. But at least now I think we're getting some reasonable sense of some of the real basics behind it. So with that, why don't I turn it back to Zalman? And thank you for that way too generous introduction. But let's go.
All right. What lessons were learned or understandings developed in the fight versus HIV that are being applied now in a race for vaccine and therapeutics?
Yeah, so I'm actually going to add a little wrinkle to that question. It being applied now, not only in the race for vaccines and therapeutics, but in the race to convey to people the risks of transmission, where the risks are, and how to protect themselves. So I'm sort of going to give away my age here. I started my infectious disease fellowship in 1986. At least the second half of it, at Montefiore Hospital and Albert Einstein College in the Bronx. And I joined the first AIDS team that had been started, the first comprehensive AIDS team that had been started in the world. It was under a fellow by the name of Dr. Jerry Friedland, who still teaches at Yale now. And he had just led the world's first study of casual contact of HIV, because back in '86, for those of you who remember, there was concern about HIV being spread maybe by strap handles in subways, or by sitting on toilets, or by touching walls, or whatever. And his study that was published in the New England Journal of 1986 demonstrated that casual contact was not the driver of HIV spread. It was through sex, it was through exchanging bodily fluids, blood transfusions, and drug use mainly. But he had to deal with a whole lot of media around that time. And in watching him and learning from him, one of the things he taught me was to try to get people to focus on the epidemiologically important ways of transmission, rather than being distracted by hypothetical means of transmission that really were not important in driving an epidemic. And I see the same thing going on here in terms of messaging or confusing around messaging. This virus, this coronavirus that causes the disease called COVID, coronavirus infectious disease, is really an inhaled virus. Okay, this is a virus that's spread through the air. You breathe it in. That's the vast majority of the ways you're going to get it. Yes, it's important to wash hands. I will never say to anyone that it's not. Okay, but the kind of distraction that I learned about in HIV, to me, when I saw like somebody on TV showing people how to wash their groceries, their grocery bags when they come home, you can do it if you want, but I think if transmission has happened that way, it's so rare to be virtually undetectable. Transmission is through the air, and focusing on those situations where you'd be putting yourself or others at risk is really key. That's one of the lessons I learned from HIV. In terms of vaccines and therapeutics, I think the key lesson is really how important it is to be collaborative. The scientific community, including basic researchers, including pharma, including government, has in support of these initiatives, have really come together in important ways to facilitate the development, testing, and ultimately regulatory release of these interventions. You're muted, Zalman.
Sorry. With social distancing and masks, what are the risks on going to shul?
So when we talk of risk, the first thing I want to be clear is that there are no absolutes. There's no, you're safe here or you're not here, okay? Everything is on a scale of risk. So key to preventing the transmission of this virus is staying away from people who are infected with it, and if one has the potential of being infected with it, not putting the virus into the atmosphere, right? Not into the environment, so other people can get it. Pretty straightforward. So masks are very effective in preventing a certain degree of virus release if one were to be infected. And social distancing, classically is the 6-foot distance that people talk about, is useful to prevent against what's called droplet-mediated spread of the virus. And we're going to get into why I would be specifically say droplet, not just breathing. Kind of looking ahead, there's droplets, and then there's something called aerosolization. Those are different, and it's important to understand that difference, and I'm sure we'll need to talk about that. So to answer that question, what are the risks of going to shul? You kind of have to understand how much a mask would prevent the virus from escaping, okay, if you're wearing a mask, and also how much a mask might prevent you from inhaling the virus if you were around a virus that did escape, right? Basically until today, literally until today, the entire message was wear a mask to prevent other people from becoming infected. And there have been studies as to how protective masks are, how effective masks are in preventing that kind of escape. It's not 100%. And we're basically talking cloth masks now, the kind of the normal stuff that you put on, right? Not surgical, not N95, stuff like that. Just your basic mask you'd find everywhere. Probably the best study and this number has been kicked around, is that generally speaking, a cloth mask will prevent 70% of the viruses in someone who is infected from escaping. Well, that's good news because you've reduced the number of virions that go into the atmosphere by 70%. However, that also means that on average, 30% of the viruses that one might have if you're infected will escape into that environment. So you've certainly, by wearing a mask in shul, reduced the potential for viral contamination of the air by a significant amount. What we've learned today, actually, CDC put out a notice today saying, hey, guess what, guys? Mask wearing isn't only pikuach nefesh. Mask wearing really does help yourself as well, right? Because, and we kind of thought this, but we were just waiting for people to document it. It does prevent particularly inhalation of droplets of aerosols and droplets, and perhaps even in aerosol, viruses and droplets, and perhaps even in aerosols as well. There's some variations based on the cloth, silk, whatever, but I'm not even prepared to go into that. I'm not sure it's important. So between that and social distancing, what are the risks on going to shul? Well, they're certainly lower than they would be if everyone in shul didn't wear a mask and if everyone was packed together. Could I tell you there was no risk? No. So that's the best I can do. And the one thing that I'm going to add, and again, I think we'll talk about it a little bit more because it's super important, is that it's not just a function of what any particular individual will do that factors into calculations of risk. It's also the nature of the spread of the virus within the population you're talking about. And unfortunately, tragically, right now we are climbing an exponential wall of spread of this virus in this country. You know, as of yesterday, today they reported that yesterday there were 140,000 cases in one day of spread in this country. I wouldn't be surprised if by the end of this week, we see, or certainly early next week, we're going to see 200,000. And maybe by the end of next week, 250,000 each day, because that's what an exponential wall looks like. It's really bad. So as this spreads, the risk of being around someone who might be infected increases, thereby increasing the risk of being in a situation where you're around people, even if you're wearing masks, even if you're social distancing. So best answer I can give you.
Okay. Thank you. All right. I would like to understand, I'm just reading this as written. I would like to understand why some viruses seem to just go away for no reason. For example, what happened in 1918? What happened to Zika? What happened to what I think was called a SARS virus of a few years ago? I don't know if the swine flu is another name for the SARS virus of a few years ago. If it isn't, it also seems like the swine flu just went away. Is it possible that by the time the COVID-19 vaccines are ready, that COVID might also just go away?
Okay, so I'm going to take the end of that question first, and the answer is no. Okay, that by the time the vaccines are ready, and obviously I'm sure we all know that the Pfizer vaccine had some great news yesterday, I guess. And we'll hopefully have time to talk about some of those vaccines. But unless there is a commitment to mask wearing and social distancing, we are, as Tony Fauci said, we're in for a world of hurt. And that's just going to be real until we get the vaccines. I will add that we actually do have a vaccine right now that's very effective, 70% effective on average, and that's called a mask. Mask is a vaccine in a way because it's going to prevent the spread of the virus. All right, so now backing up, well, let me just say mask, if you add social distancing, if you add ventilation, those are more components of
Take a shower.
that will make it more effective. Why do some viruses go away? Well, they don't go away for no reason. Okay, they just follow basic biological principles of pathogen spread. There's actually an equation that governs the spread of a pathogen through a population. I'm not a mathematician. Zalman, you're way closer to being a mathematician than I am. I can't, yeah, that's not for me. But I know enough about the equation to point to a certain variable in that equation that many of you have heard of, in all likelihood, based on the discussions that were happening early in the pandemic. And there's this mysterious variable called the R naught, R with a little zero. And that basically is the factor that illustrates in the equation how many people one infected person would be expected to infect, all else being equal, if you just kind of let it go, right? So the thing is that factor is an exponent. So if you have an R naught that equals 2, that means that one person will infect 2 people. 2 people will infect 4. 4 will infect 8. Okay, that is massive spread. In fact, this virus, left to its own, has an R naught of somewhere around 2.5 to 3. So it's even left to its own devices more apt to spread than even like a 10 to the second spread. And that we can illustrate, if you want to talk about it later, that's a fairly dramatic rate of spread. Now, the key to controlling the spread of a pathogen like this particular coronavirus is to do what you can to lower the R naught below 1. Because if you go below 1, that means one person will infect fewer than one other person, which means ultimately it's going to die out. And the lower you can get that R naught, the more quickly it's going to die out. Well, how do you reduce the R naught? You get people to wear masks. You get people to social distance. You vaccinate people. And by the way, people become infected, and those who survive develop antibodies that for a certain amount of time remains unknown, will protect them, or should protect them, from becoming reinfected. So in the case of the 1918 flu, really it was the latter case. I mean, there was no control, there were no flu vaccines. It was just, virtually the entire world becoming infected with this terrible pandemic, H1N1 flu, that killed somewhere between 50 and 100 million people. It was the worst single infectious catastrophe in the history of documented history of the world. With Zika, it just kind of burned itself out. I can't give you a good explanation for it. I'm not exactly sure with Zika. With SARS is caused by a coronavirus. What happened there was just like COVID, by the way, which is a coronavirus, a different related. That's why the actual name of this particular disease is SARS-CoV-2, because it's the SARS-like coronavirus. It's not exactly like SARS, but it's like enough to carry the name. Through a aggressive efforts to identify infected people and contacts of infected people, and then, quarantining, which is sort of an extreme of social distance in a way. You get the idea. I should have added the importance of quarantining, testing, contact tracing, et cetera, in these efforts to reduce the R naught. That there was a very aggressive global public health response to both SARS and to SARS, and that took care of that. Swine flu is not another name for SARS. Swine flu is influenza. It's not. It's not. Coronavirus. But there we had vaccination, and between vaccination, between herd immunity, and just evolution of the virus, eventually that these viruses, influenza is a whole different topic. It's a fascinating topic of seasonal flu versus pandemic flu, and it's something we could spend a whole lot of time talking about. But basically, what happens is that a pandemic influenza eventually becomes the new seasonal influenza, and then you get vaccinated for it every year, and that's what happens.
Okay. We're going to take one more question from the ones that were submitted beforehand, and then we're going to go to the chat. We have four questions in chat already.
Okay.
So that's our game plan right now. Have you heard anything about Dr. Christiane Northrup with nanotechnology in vaccine tracking biometric data?
Okay, so I think we should go to the next question. This is not going to take long to answer. I had never heard of Christiane Northrup. I looked her up when you sent this to me. And, she's an OBGYN who basically has had her tweets and communications removed from Facebook and removed from Twitter because she's out there saying that the vaccines have not been placebo controlled, which is blatantly wrong. So I would say enough said about Christiane Northrup.
All right. We're in the chat now. So these were just asked since the meeting started even. I would like to ask you a question regarding vaccines. Since vaccines are tested on healthy people, how does a person with diabetes or with multiple autoimmune disorders know it's safe for them to use?
So it's a great question. And you have to get into the kind of vaccines that might raise real issues for people with diabetes, autoimmune diseases, immunosuppression versus those that are highly unlikely to do that. So there are many different vaccination strategies that researchers, pharma uses to come up with vaccines that can protect against all kinds of different infectious diseases. We're seeing that as in terms of the response to coronavirus or COVID. You're seeing that. It's a great illustration. The kind of jargon in the field is we call them vaccine platforms, right? Strategies, platforms. So the first vaccines that were ever created back, in the turn of the 19th to the 20th century, Pasteur and those guys, they were using live attenuated vaccines, which basically means you take the organism that causes disease and you weaken it. You might weaken it through exposure to, some chemicals, oxidating chemicals, or, whatever. There are all kinds of techniques to, formalin. You can weaken but not kill them. And those, there are all kinds of variations on live attenuated. Again, that's a whole nother lecture. But those vaccines are safe after being proven safe by studies now, through FDA observation. For the record, I used to work in the vaccine regulatory group at the FDA, so I spent 10 years doing this. They're safe under most circumstances, but in some people, they may not be safe because the safety is based on an appropriate immune response. So if you are immunosuppressed, you're not supposed to get, if the smallpox vaccine, for example, were still being used, and it's not, thank goodness. But if you're immune suppressed, you're not supposed to get it, because even though it's relatively safe, it can kill you if you're immune suppressed. In terms of autoimmunity, there are certain ways to what would be the word for it, to heighten the effect of certain vaccines through additional chemicals called adjuvants. There's been a theoretical concern about enhancing, potentially some tie with autoimmunity. I remember having reviewed this at the FDA. We haven't found any evidence of that, for the record. But theoretically, there are some people who worry about that. The vaccines that, like the Pfizer, and by the way, again, it's not. There's never absolutes, and there's always a risk benefit that someone has to make when you're choosing a vaccine, what vaccine you're going to use based on who you are and what your potential risks are. Right off the bat, the vaccines that are leading the charge in COVID should be among the safest, if not the safest, vaccines that have ever been made. They're a brand new technology. They're called messenger RNA or mRNA technology. They're not live attenuated. They don't have adjuvanted proteins. Not to say adjuvanted proteins are dangerous, because generally they're not. They're not whole killed. Okay, that you're not taking the virus and killing it. You're basically taking a piece of genetic material that codes for the protein that you want to develop an immune response against. And researchers have figured out how to deliver that little piece of genetic material to your own body in a way that allows your body to, on its own, make the protein in its own cells. So it's extraordinarily safe. The question really was, would it be effective? Because it's never been done before. There are no licensed mRNA vaccines out there. Well, Pfizer's vaccine, that's done with a German company, BioNTech, just showed more than 90% efficacy in their phase 3 trial, which is amazing. I personally never expected that. Right behind them is Moderna, and they're called Moderna because it's MODRNA, Moderna RNA vaccines, right? There's a third company, T-Bio, that's coming out with an RNA vaccine. So these vaccines should be very safe. There's going to be others with other platforms. There's going to be an adjuvanted protein. I think Novavax is probably the first out of the gate with that. That should be safe. But again, it's always risk-benefit balancing. And given the safety profiles of these vaccines versus the potential risk of acquiring the virus, at least from my perspective, the balance goes to taking the vaccines.
Okay, thank you. Are there any contraindications to the vaccine, such as Epstein-Barr, mononucleosis, or a history of serum sickness?
No, so serum sickness is not relevant to this. That's ancient technology, and EBV, there should not be, no.
Okay. Yeah. Okay. What are the chances? And this is a tough one because I just read about this with the Denmark thing. What are the chances of mutant viruses developing before a COVID-19 vaccine is developed?
So, I can't give an absolute answer to that. I've spoken to friends of mine who are researchers at the NIH about this. Their opinion is that it's very, very low. That, all viruses mutate, and this one's mutating. I mean, you can see differences in virus. You can track the viruses from China. You can track viruses from Europe. By the way, the United States was infected with the European virus, not a Chinese virus. So issues around China viruses is somewhat irrelevant or not on point. But the mutations are more markers than they are then they've proven capable of modifying the immunological face that the virus presents to the body. So there has not yet been a mutation that I'm aware of or that my friends are aware of that has changed that immunological profile in a way such that, for example, if one person were to become infected with a virus from Europe, develop appropriate levels of antibodies that are protective, then gets exposed to a virus from China or a mutated virus from Europe, they wouldn't be able to control that virus. No one can promise that won't happen, again, but I think we're in the realm of the theoretical when we talk about that rather than the real. And the real right now and for the foreseeable future is that when we get a vaccine that protects against the virus we have circulating here, it's going to protect against the viruses that will be circulating everywhere. Now, and for long periods of time. That's what we think. That doesn't mean that people won't be testing to be sure that's the case, or not, that we're not wrong about that. People are aware of the potential, and it's going to be part of everyone, every broader public health responsibility to assess that possibility.
Okay, I'm going to go a little out of order because there's a question in the chat that's about that specifically. Someone asked if you can go a little more into the China virus versus the European virus when talking about COVID-19. Because that was the first I've heard of that also. Like, what's?
I can't. I mean, it's just a question of saying that you can trace the viruses based on small mutations in their genes to say, okay, this one looks like it comes from, this one has a specific little difference in a couple of base pairs in its, in its genetic material, as compared to this one showing the signature coming from Europe, Italy, France, and these genetic researchers can actually get fairly specific using kind of genetic epidemiological techniques. My only point was to say that even though there are these tiny little mutations that are useful when tracking the virus epidemiologically, in terms of where the source is, where it's going, et cetera, these changes, at least until now, are not relevant when it comes to assessing they have not been significant in changing the immunological face of the virus against which your immune response or the immune response engendered by a vaccine will protect against. That's really my only point.
So Bezrat Hashem, the Pfizer vaccine would work against China's little version or Europe's version in theory.
That is the hypothesis. That's what will be tested. And everyone expects the answer to be yes.
Okay. I have one here, and then I have to look at my email for one that somebody said. Okay, so will these first vaccines from Pfizer and Moderna be recommended for children also?
I think so. I think so. I don't know exactly what's going to be on the label. This depends on who was enrolled in the trials and how broad the FDA regulatory group will feel comfortable in terms of broadening the age range. Usually the initial, when you're doing vaccine regulation and you're writing the label, you're giving authorization for use, you only approve for the people, for the age groups for whom that were actually enrolled in the phase 3 clinical trial. So it's an interesting question. I don't know the answer to that. I don't know the age ranges. I'm sorry. I wish I had the information. I don't know the age ranges enrolled in the trial. I don't know how flexible the FDA might be around this. I'm going to guess that, again, since one of the things about FDA licensing you have to understand is everything there, like everything we're talking about today, is risk benefit balancing. Everything is risk benefit. There's no absolutes. And they may say, okay, maybe they only enrolled sort of the classic population is 18 to 55, but they may say, look, there's absolutely no evidence of any important adverse events, if that's true, from the data, from the phase 3 safety data, and all the data that came before phase 3. And given the risk of the infection and given the importance of protecting as wide a population as possible, they may grant use in younger kids. I don't know if it'll be starting at 2, maybe it'll start at age 6. Maybe it'll start at age 12. So I guess what I'm doing is guessing. And we'll see what the FDA decides as they negotiate this with the drug companies. But that is, I'm sure, on the table.
Okay. This is all right. So this just came into my email. There are so many anecdotal stories from Monroe, New York, from Marseille, from a nursing home in Texas, and in Muncie from California, that hydrochloroquine with Zithromax with zinc, when given early onset, prevents the disease from becoming serious. Has this been definitively studied?
It's been studied, and I can't quote you the studies, but I have seen the reports of the studies, and it doesn't work. The key word you use there is anecdotal. We don't license things when as if I'm part of the FDA now, at least I kind of guess you keep it with you even when you leave. We don't license things based on anecdotal data. You need carefully controlled, placebo-controlled, blinded trials to make that kind of assessment. And there are no data to suggest that hydroxychloroquine works. It's a dangerous drug. It affects cardiac rhythms, and I wouldn't use it. I wouldn't let any of my family have it. If I were running a hospital, I wouldn't let it on the formulary for this kind of thing. It's not what you want. And by the way, there are interventions that are being developed that are showing that they do work. Dexamethasone, it's all over that. I mean, it's just a steroid, but dexamethasone really works in severe disease. I'm not sure about remdesivir. That's just an antiviral. Set that aside. Monoclonal antibodies that they're developing now. One was just licensed for the first time, I think today or yesterday, by Lilly's monoclonal, which is the one that Chris Christie got. Works is being not licensed but approved under emergency use authorization for a certain situation, not for everybody who's infected with COVID, but certain situations we could talk about. Regeneron's monoclonal is probably going to be approved in the next week or two. That's the one that Trump got. There's a third company making monoclonals called Vir. They won't be ready probably until a few months from now. So there are interventions, but hydroxychloroquine and azithromycin isn't one of them.
Okay. Follow up from the other question before. Have people over 65 been, as far as you know, been included in these phase 3 trials?
I don't know. I don't know. Well, no, I do know. Okay. Well, I know they've been included in trials because I tried to enroll in one of them. Um, uh, but whether they're in the phase 3, I'm not sure. Okay, I'm not sure. The reason that would be relevant wouldn't be so much from the safety perspective as from the efficacy perspective, because as you get older, your immune response tends not to be as robust as in younger people. And so, like in influenza, those of you who are over 65, they'll say, okay, well, now you need the vaccine for the older people, which is basically a higher dose than given to younger people. So that may be of issue in the COVID, in the mRNA vaccines and others. I don't know if they ferreted that out yet, though.
I'm glad people are still throwing questions in chat. Are there any animal products in the vaccine, such as LAL from horseshoe crabs or non-kosher animals?
Not that I know of. I mean, the mRNA has a fat droplet. It's kind of a nano technology that allows it to go in, as I understand it, allows it to be absorbed and taken up into your cells. Adjuvanted protein. For the adjuvanted protein, some adjuvants use material from shark. So that's a non-kosher animal. But I guess I'd put it to your rabbis is to say, is that even relevant? Because if you're talking about a medical intervention, my learning, which is certainly not to the level of yours, but my level is that doesn't matter.
Okay. Before I go, we have about 4 more questions from the pre-list. But before that, there may be people that aren't good at like using. Oh, wait, there's another one in chat. Have they tested the vaccine with some people who already had the virus? Will that matter? So that goes with one of the questions on this is about people getting it a second time.
You don't want to test the virus. There's no reason to test the vaccine on someone who's already been infected because they've already developed an immune response to it. So no, that would not be mainstream. I mean, there may be some doc somewhere who decides to do it and get a paper published, but it's not an important public health goal. In terms of whether someone can be reinfected with COVID, again, I don't want to be absolute about it. I think that, what is apparent now is that is not likely to happen after you've been infected because you develop antibodies to it. And those antibodies should be protective. It's one of the reasons the virus goes away. It is the reason the virus goes away after you've been infected for those people who actually recover, right? Their antibodies basically wrapped up the virus and killed its ability to replicate before it killed you. Those antibodies don't just go away, they stay. Now, how long they're going to stay, we don't know. Antibodies are supposed to go away over time. You don't want your body filled with your blood filled with all the antibodies from every pathogen you'd ever seen in your life. It'll turn into glop. It won't circulate. Okay, so antibodies have to wane over time. That's what they're supposed to do. But there's supposed to be, in an effective immune response, a memory response, so that if you were to be exposed later, the antibody response would come back up again, okay, and protect you very quickly before the virus had a chance to replicate and actually do damage. We think that's going to be the case with people infected with COVID. People who have been infected with COVID are being followed for that, to be sure. Are there situations where that may not be the case? Well, yeah. If someone got infected and then got immunosuppressed for some reason, took chemo, or got bad sick for some other reason. What would happen if someone had been infected, developed antibodies, but then, was a doc, say, and went into an ER that was overrun with COVID and wasn't wearing a mask? That wouldn't happen, but this is just a hypothetical. My point being that, would it be theoretically possible that someone getting huge exposures to the virus would be at risk for reacquiring it even if that person had a certain protective antibody level? I don't know. It's theoretically possible. So you don't want to. So even though the message I think from that is that even though you've been infected, you don't want to be cavalier, assuming that no matter what you do, you can't reacquire. It may be the case that you can't, but it's prudent not to make that assumption.
The follow-up to that was, so would you recommend that someone who had got infected with COVID does not take the vaccine?
I personally don't see the reason why someone who's been infected with COVID would be vaccinated for it. That's correct. Maybe somebody, but I don't want to be the last word on this. I'm just, if this is a pop quiz, I'm saying no, they don't get infected, they don't take the vaccine. If the CDC comes out and says you're wrong, they should, well, then I'll learn something, but I don't understand. I don't understand why they'd say it. I just like to see the rationale. I wouldn't expect, particularly you're also going to be in a situation where you're going to be trying to get the vaccine to the most, the people at greatest risk. And the people who have already been infected and develop antibody responses are going to be the end of that line. And I don't think they're even going to be in the line. So
if one has had a Guillain-Barré syndrome response, would it be recommended?
Oh man, these are really hard questions. I'm going to pass on that one. I don't think it would be a problem. That would be my guess. But there I would double check.
Okay. Why are states requiring a 2-week quarantine when returning from out of town if a person waited a few days upon returning home, got tested, and was negative? Are the tests just not fully accurate?
Yeah, the tests are not fully accurate. The states are trying to be very careful, especially now, as I've said, when we're climbing the wall of exponential spread. So I think it's just erring on the side of caution.
Do you think there will be a national mandate for the vaccine?
I don't know. There's so much polarization. There's so much anti-vaccine sentiment out there. I think there will be a national a message to strongly suggest that if it's available, one would be smart to take it, but to actually force someone to take it, I'm going to have to punt on that. I really don't know how that policy is going to evolve.
Okay. We're back on the sheet. There's only a few more, and we're getting close to our time officially, whatever. Which COVID tests work? Are there different levels of accuracy?
Okay, and this is what I'm particularly not qualified to to respond or suggest. I was saying that the diagnostic test is really not in my wheelhouse. I can tell you that there are a lot of tests out there. There are a lot of different types of tests. There's the PCR test, the polymerase chain reaction, which tends to be the most accurate. There's the antigen test that is less accurate but easier to administer and cheaper. Those are your two key tests to determine whether or not you're actively infected. There's the antibody test and when I say the antibody test, there are a lot of different companies making antibody tests. There are a lot of different companies doing PCR. There are a lot of different companies doing antigen, which is why it's a mess. And the FDA has not been particularly. The FDA and the CDC have had issues in trying to regulate them. So I don't think that there's a really good solid way, at least that I have access to, or I'm not aware of, to say, okay, well, this is the best, and this is so the best I can do is say, all right, you got those two species of tests to test if you're infected. You've got the antibody assay to test whether you've been infected and have developed antibodies, and that's pretty much it. And I'm reading the rest of the question. I think that's probably the best I can say about tests.
Okay. Once the vaccine is out and folks start getting it, are we immediately safe? Can people who have not been attending start going to shul?
So I think that's a really important question. I'm glad that question came up, because it's the exact same question that came up when I was talking to the artistic directors. Yeah, kind of to change the question a little bit, I think, what's the metric for assessing potential safety for everyone getting back together again, like we all want to do? Unfortunately, the amount of vaccine usage is not necessarily going to be the metric. The metric is going to be the incidence of disease in the population, the incidence of infection. So when those rates start to come down and really bottom out near zero, that's when it will be safer for people to come together in shuls, in theaters, in indoor stadiums, etc. So that's what you got to look for. And by the way, it gets back to the importance of masks, because it's not, if a community really wants to put itself in a position to start gathering again, then basically you got to do all of the above, and everybody needs to do it. Everybody needs to do it. It's not a political issue. It is a biological issue. So what do? You wear a mask when you're in public, when you're around other people. If you're walking down the sidewalk and you're taking a walk and no one, you don't have to wear a mask. It's fine. You're not going to catch the mask from the air. But if you're around people, you need it. You need to do it for them, and you need to do it for you. You need to do it for your community. You put the mask, takes masks seriously, takes social distancing seriously, avoid gathering in large numbers, even if among your family. Why? Because you're increasing the risk that somebody in that group may have accidentally been infected. Maybe they're asymptomatically infected. Maybe they're pre-symptomatically infected. But the more people you allow into your little bubble, the higher your risk is. So you mask, social distance, get together if you do in well-ventilated outdoor spaces, and then get vaccinated. When you do all that, if you do all that as a community, you will be ahead of the curve and you will be able to gather sooner.
Okay, we got two more these and one or two more in the chat. What does it? All right. When is it safe to take the vaccine? Do you think the Pfizer vaccine is good? How long would you wait to take it?
I would take the Pfizer vaccine tonight. So, again, you kind of look at you look at what the platform is. You look at what the nature of the serious adverse events, the adverse event profile is from the data, the phase 3 data. You look at the biologically plausible adverse events, and like I just said, I can give you a whole list of biologically plausible adverse events from a live attenuated vaccine. Not to say I wouldn't take one, measles, mumps, rubella, they're live attenuated, but they work. Okay. With this one, I would take it. I'm ready to roll up my sleeve as I'm talking to you. 90+% efficacy with that kind of profile, safety profile, you got to take this vaccine. Given everything we know now, and given the plausibility, and given the risk of acquiring COVID.
All right, last paper question, and then chat, and then I think that we'll call it a night. A lot has changed since we first learned about COVID. when folks were recommended that people wash their groceries in quarantine, we mentioned that before, and quarantine their mail. What do we know now about this, how this is transmitted that we didn't know at the beginning? And what's it mean that it's transmitted in aerosolized manner? And what's this mean for being indoors?
Yeah, so this is what I sort of hinted at early in our conversation. I think at the very beginning, people were really focusing on kind of what we call fomate transfer, where you go from fingers to nose, as well as droplets. And so I'm going to set aside the surface transfer again. It's not that it's not possible, but it's not the important means that is driving the pandemic. The important means is in the air. So I think that's one of the things we learned early. All right. I mean, we've learned since the early days. Now, the other thing that's really important we've learned is that we've always known that there broadly speaking, two ways that one can, how can I say this discreetly? That one can throw spit out into the world. Okay, sorry, but that's the way it is. One is through droplets, and the other is through aerosol. Now, when you think of droplets, think of, what would be a good analogy? All right, how about this one? You've been to a fireworks display and you've seen fireworks on the ground and the things spurt out and they come down in like this umbrella. And they don't fall very far from that central source of the fireworks display, kind of like a Roman candle, right? Those are droplets. They go up, they go out an umbrella, and they fall in this fairly discrete diameter, right? The wind really doesn't take them. That's where your 6 foot recommendation comes from. 6 feet, 2 meters, all about if someone is talking, like I just said, a T, and I'm throwing a droplet that I can actually see on my screen. If you're davening, if you're singing, if you're whatever, you're projecting these droplets for the most part. Now, what we've learned since then is that this other component of how you throw stuff out of your mouth, nose, mouth, into the world is also really important, and that's aerosol. Aerosols are particles that are way, way, way smaller than droplets. When you think of aerosol, if you're going to use the fireworks display, think of the big fireworks that go up way high in the sky. The other way to think about aerosol is, if you open the shades in the morning and you see the light coming in and you see the dust particles kind of floating there, right? You've seen that. That's aerosol, okay? They float, and they can float for long distances. And pretty early in this pandemic, it was pretty clear that people beyond that 6 foot radius that we talk about social distancing were getting infected. Like in a restaurant where, depending on the direction of the air and the ventilation system, people downwind, tables downwind, there were people like 28, 30 feet getting infected, where people upwind 5 feet weren't. That's not droplets. Those are aerosol. There are a number of other examples that occurred like this. So what we've learned, and I think this is super important, is that when you're indoors and you have particularly, you have limited ventilation, even when the ventilation isn't bad, you still, if you're not really replenishing, if you have sort of your classic indoor ventilation situation, then aerosolization can occur. And in aerosolization, forget about 6 foot. It can be 30 feet. Okay. Um, and the other thing about an aerosol is that even though each of those little droplets doesn't carry as much virus as in a droplet, right? They tend to go deeper into your respiratory system. So a droplet, if you inhale it, most of them actually get filtered out through turbulence in your nose, your throat. You know, we're built to protect ourselves, our lungs, against pathogens delivered that way. Aerosols are more efficient at escaping that type of protection and are more likely to deliver the virus directly into your lung. And that ain't good. That increases the chance of having a more serious outcome. So I think that's an important message. It really emphasizes that to a certain great extent, this is an indoor disease. This is a disease that's spread inside. When you're outside, literally the sky's the limit. You don't have aerosol clouds that just hang around. And by the way, the other factor, I mean, it's complicated when you're talking about what are the factors that result in higher risk of acquisition. But one of the things, the two other factors I really haven't mentioned are duration of exposure. So like if you're walking outside and someone coughs who's coronavirus infected and you walk by them, and even if they're not wearing a mask and you're not wearing a mask and you kind of walk through that little cloud of that one little exposure is highly unlikely to result in you becoming infected. Whereas if you're sitting in a room and you're breathing in, breathing out, I think the CDC talks about prolonged, and I forgot the number, is it 3 to 5 minutes, 2 to 5 minutes, something like that. So it's not just walking through. The other, and it's a component, sort of a corollary of that is the dose, the amount of virus you're actually inhaling. So putting all those factors together, being outdoors, just being outdoors will hugely reduce one's risk of acquiring coronavirus infection.
If one has had neurological reaction to a vaccine, would you say it is dangerous again? And then also with that, it said, please discuss herd immunity.
I would look, a neurological response to one vaccine platform does not suggest that you're going to get one from a different vaccine platform. That being said, I'm gonna so my gut feeling is that, again, with if you're talking about a vaccine that's based on a totally different platform, the likelihood is it's going to be safe. However, I think that before you do choose to take the vaccine, it's something whoever's writing this question needs to discuss with his or her personal physician. The likelihood is that that person's not going to be able to answer your question and may have to do some digging, may have to call, send a note to the FDA or the CDC or somebody to get some clarification. But the likelihood is it's going to be okay. And in terms of herd immunity, it's not that complicated. Basically, I like to come up with analogies. I guess here an analogy would be a forest fire, right? I'd have to kind of tweak it a little bit, but if you got a forest fire burning and you see trees left and right, going up in smoke, and if you, in this hypothetical, if you came up with a chemical that you could douse the surrounding forest with that was a flame retardant and wouldn't allow the surrounding fire to escape the area that already was burning, okay? Well, then the fire would eventually die out because the embers would fly into the area that was protected by the flame retardant but they just flame out and go away. That's herd immunity. You have enough people who are protected that don't allow the spread of the virus to occur. Put another way, you have enough people who develop immunological protection so that the R naught goes way below 1. And so one person may only, maybe 0.1, which, by the way, is a reason that when we do have a vaccine, it is important from a community perspective for people to take it. Because if you don't take it, then you might be one of the unprotected trees in that forest, which means that the trees around you are protected, but then the ember could land on you, poof, you get infected, and then it can spread and hopscotch to another population that's not yet protected. So this is really a kind of a community bond. The reason you don't want it to happen naturally, Scott Atlas is saying, "Oh, let everybody get sick and we'll develop herd immunity." That is beyond irresponsible. That is breathtakingly, literally breathtakingly irresponsible. Because the other thing we haven't mentioned, and I know we're I don't know what our time is now, but we don't know everything about this virus yet. We don't know the full picture of this virus's pathology. There are microthrombi, that is small clots that occur in a certain percentage of the people who become infected. There is myocarditis, that is an inflammation of the heart muscle that occur in a certain real percentage of the people who are infected, including young people, including young athletes. Now, most of them seem to get better. Hopefully they'll remain better. Can we guarantee that? No. So, we don't want to have a population develop herd immunity through wanton spread of a virus when we do not fully understand the clinical implications, particularly the longer term and the shorter term disaster that this results in when you're talking about hospitalization. I mean, even now, hospitals are now overflowing again. They've got field hospitals out in El Paso. I've got family members in Pennsylvania now who are saying our hospitals are fill up again. And it's going to get bad.
Two more from chat, and then we'll call it. If the US was infected via Europe, how was it transmitted to Europe?
How did it go? That's an interesting question. I think it did come initially from Wuhan, okay? But the virus that came from Wuhan into Europe then had a small mutation that distinguished it from the one that stayed and spread throughout China. So the initial nidus was China.
All right, and this one I wanted to ask before when you were talking about, because it's the same topic, but isn't it now being said that we should do more than 6 foot distancing?
I'm saying we should do more than 6 foot distancing, and I think that is the case indoors.
6 or more?
More.
Okay.
How
much more?
There's so many variables. It's just hard to say. I mean, for me, I don't know. I think more is, what are you willing to do indoors and why? I am not willing to, for example, eat in a restaurant indoors. I'm just not. I don't need to. I'm not willing to take my mask off. And, you know, even, and maybe the people are 10 feet away or 12 feet away on a table. It's not, it's risk benefit balancing. Why am I there? Is it worth the risk to engage in the behavior? That's what you got to ask yourself. You know, would I eat outdoors within 6 feet? I mean, outside, you know, with a 6 foot distance? Yeah, maybe. Yeah, I've done it. You know, I mean, because some friends of mine wanted. I didn't run to do it, but I did it. You know, because outdoors. You know, I'm recording this, and some public health guy's going to get this and start yelling at me, but I hope it's not the case. You know, aerosolization really isn't that big a deal. It's not as big a worry as it is indoors for all the factors I've talked to you about. You know, density of virus, length of exposure, etc. Indoors, it's different. So indoors, I basically don't do it except to do grocery shopping. And I'm not nervous when I grocery shop. I mean, I'm wearing a mask and everyone's wearing a mask. I live in Maryland. Everybody kind of does what they're supposed to do, luckily here. There's no pushing back. We shop, and then, you know, when I have friends over, we go out on our back porch if I don't really know them well. And if we do eat indoors, which is with other people, which is very unusual, I you know, I can't, you know, maybe 5 times since the beginning of the pandemic, I've known these people really well, and we all trust each other. But we don't go more than 6 people.
Haircut?
Haircut? I about 6 weeks ago, I got my first. Actually, my wife was cutting my hair for a while, which was good. She did a good job. But I kind of felt, frankly, I felt bad for the person who's cutting my hair. Her business was horrible, and I said, look, I'll go back and do it. They were really careful. They only let 2 people in the store at a time, and they're all masked, and they have all kinds of stuff on. The one question, by the way, that was on here is also the face shield versus the cloth mask, because my hair cutter asked me this actually, and it's the mask that's important. The face shield really does not give meaningful protection.
Somebody asked that this is going to be this won't be a yearly vaccine, right? This would be like basically once. It might be a series or something, but
I don't know. I don't think anybody knows. I think, you know, we're hoping we're hoping that once we get everybody vaccinated, number one, we're hoping it's just a one-time deal. It's not like influenza. We don't think it's like influenza. We don't think that there are going to be changes to the genetics of the virus that actually change that immunological phase. See, that's the problem with flu. I keep talking about immunological phase, and in influenza, the small changes that result in seasonal flu and the big changes that result in pandemic flu change the immunological face of the virus. With this one, odds are that that's not going to happen. And hopefully one series of vaccinations will do it. And I say that because understand that for the mRNA vaccines, at least, you're going to need 2 vaccinations a month apart. So.
Dr. Schrager, it has been an honor and a pleasure. Thank you so much. Thank you so much. God should bless you and your family with continued health and all the good things you're bringing out to us. It should come to you also. You should be blessed forever. Thank you so much.
Thank you. No, I really appreciate it, Zalman. Thanks for the opportunity. And I wish everyone health and happiness, and just be careful and do the right thing. You know, everyone's trying to do their best.
Thank you so much for your time and answering our questions.
No, it's my pleasure. Thank you. All right. Be well, everybody.
Thank you, cousin.
Yeah. See you.
Thanks, cousin.
Bye.
Toda rabah.
There's a video with the thing that I put on my phone. I watched it. What this resource is it?